This experiment aims to assemble nucleosomal arrays that contain a series of tandemly repeated nucleosome positioning DNA sequences with an equal number of nucleosomes. First, refold histone complexes by dialysing the core histones out of denaturing buffer. Then, separate the histone octamer using a size exclusion column. Next, combine the histone octamer with template DNA at a range of molar ratios and deposit on the DNA as nucleosomes by step dialysis from high to low salt. Next, test the samples by sedimentation velocity to determine the proper molar ratio for the desired level of saturation. Results can show the level of saturation of the nucleosomal arrays based on their migration through solution under centrifugal force or based on an atomic force microscopy generated image.
The main advantage of nucleosomal arrays, rather than mononucleosomes, is that the nucleosomes are connected by linker DNA, imposing an additional structural constraint.
Though this method can provide insights into chromatin structure, it can also be applied to other systems, such as in vitro transcription study on chromatin templates.
Ryan and Anna will now demonstrate the assembly of nucleosomal arrays from recombinant core histones and nucleosome positioning DNA.
Begin with the purified lyophilized recombinant core histone proteins H2A, H2B, H3, and H4. Dissolve approximately 5 mg of each in 3 ml of unfolding buffer, and incubate for at least 1 hour at room temperature with agitation.